Bacterial conjugation is a process of genetic transfer between bacterial cells that requires direct contact between the cells. Many, but not all, species of bacteria can conjugate. Conjugation can occur between cells of the same species or even between cells of two different species. A small DNA circle or plasmid called the F factor is required for conjugation. The F factor stands for fertility factor. Strains of bacteria containing the F factor are called F+. Those without it are called F-. An F+ cell, or donor, produces a structure called a pilus to connect with another recipient cell. To begin conjugation, the F factor is cut at a specific region called the origin of transfer by a protein assembly called the relaxosome, which associates with the strand to be transferred, or the tDNA strand. Accessory proteins of the relaxosome are released, but a portion of the relaxosome called the relaxase remains attached to the tDNA. This tDNA relaxase complex is recognized by a coupling factor and transferred to the exporter, a complex in the F+ cell that is contiguous with the pilus. The exporter pumps the tDNA relaxase complex into the recipient cell. Once the entire tDNA molecule is transferred to the recipient cell, relaxase joins the ends to make a circular DNA molecule. As the tDNA is transferred to the recipient cell, it is replicated to become double-stranded. In the donor cell, the F factor DNA was also replicated to become double-stranded. This actually occurred as the tDNA was being transferred to the recipient cell. In the end, both cells wind up with a complete double-stranded copy of the F factor. Their connection through the pilus is released, and each is now an F+ cell that can go on to conjugate with other cells.